Dracunculiasis, also known as the Guinea worm disease, is caused by the nematode roundworm Dracunculus medinensis. The Dracunculus medinensis is found primarily in the tropical areas of Asia, Africa, the West Indies, and South America. There are many structures on the anterior end of the worm, such as the mouth, which aids in consuming gastric juices of the host, the muscular esophagus, the glandular esophagus, the cervical papilla, and the nerve ring. The Guinea worm disease causes a variety of symptoms, such as blisters, hives, ulcers, diarrhea, nausea, vomiting, fever, itching, and oedema. While there is no specific treatment to treat the disease, nor is there a vaccine to prevent infection, the worm can be removed once developed. After the worm emerges, it can be pulled out a few centimeters per day by twisting the worm around a support such as gauze or a stick. Generally, this process takes a couple of weeks, and it comes with the possibility of secondary bacterial infections and other issues if the entire worm is not removed. The drugs metronidazole and thiopentazole should be administered to treat the infections caused by the worm, and additional anti-inflammatory medicines and antibiotic ointments can be used to reduce pain and further infections. The nematode is acquired through drinking contaminated water that is infected with water fleas housing the larval stage of the nematode. The ingested flea contains mature worm larvae that are in their third stage of development. When in the stomach, the gastric juices break down the water fleas, and the released larvae travel from the intestinal tract to the abdominal tissues. They grow and reproduce at the connective tissues, and the fertilized females move to various parts of the body, while the males die upon mating. After a year of development inside the body, the worm breaks through the skin, forming a blister. After emerging from the primary host, the worm releases millions of first-stage larvae, and blister fluid is secreted. The larvae laid in the water have a chance of being consumed by a host, in which they can develop into the later life stages. However, if the larvae do not find a host in 3 days, the larvae dies. Once the larvae are ingested by the intermediate host, such as the water flea, they molt twice and reach the third stage. This form is capable of infecting a principal definitive host, such as humans, dogs, and other mammals.
Innate immunity is the first line of defense in human body. When the water flea containing the larva of the guinea worm is ingested along with the intake of contaminated water, the first line of defense it encounters is the mucous membrane of the digestive tract. The mucous membrane serves as sort of a sticky tape which traps passing invaders and moves them along the digestive tract to the stomach. Stomach's gastric juice may also act as an innate immunity by creating a hostile environment for the water flea. When the water flea encounters the gastric juice present, the flea gets dissolved due to the low pH of the stomach. However, the larva is able to survive the low pH and moves on with the rest of the digestive food towards the small intestine. Once the larva gets to the small intestine, it slips past the lining of the small intestine and lodges itself between the smooth muscles that are present there. Once the larva is embedded in the tissue, several TLR5 receptors present in the surrounding area are triggered and bind to the flagella of the larva in order to inhibit its movement. Next, the macrophages that are present in the tissue of the intestine also recognize the larva as foreign and work to engulf it. However, due to the size of the larva, the macrophages are unable to destroy it. Further, the macrophages do release histamine, which causes the blood vessels to dilate and immune cells to travel from bloodstream to the surrounding tissue. In addition to the macrophages, mast cells also release histamine. Eosinophils present in the tissue are hardwired to attack parasites like the guinea worm larva. They produce destructive enzymes in order to combat the parasite. Next, the complement system kicks in. This system consists of many proteins that circulate the blood in an inactive form, but become active when they encounter the pathogen. When the proteins are activated by the molecules present on the surface of the larva, they produce a cascade of biomechanical reactions that push the larva towards lysis. In addition to triggering the complement system, antigens present on the surface of the larva also help elicit a response from nearby B and helper T cells. When the antigen-specific antibodies present on the surface of B cells successfully bind to the epitope of antigen present on the larva, the B cells are activated. Once activated, the B cells quickly replicate into more effector and memory cells. The effector cells produce water-soluble antibodies that can then attach to the antigens on the larva. Once attached to the antigen, the antibodies work by attracting the attention of macrophages towards the larva. The activated B cells also engulf part of the antigen presented on the larva and then display it on MHC2 molecules. The MHC2 molecule works in conjunction with helper T cells. Specific antibodies on helper T cells can recognize the MHC2 molecule and activate the cells. The activated helper T cells replicate and then produce cytokines, which help B cells replicate faster. Along with the effector B cells and helper T cells, memory B cells and helper T cells are also produced, which last well after the infection is gone and provide lasting immunity from the larva of a guinea worm. Helper T cells and B cells are part of what is known as the humoral immune response. Additional immune cells, such as cytotoxic T cells, are part of the cell-mediated immune response. While B cells attack foreign pathogens, which in this case is the guinea worm larva, cytotoxic T cells attack infected host cells by forcing them to undergo apoptosis. Cytotoxic T cells do not play an active role in the guinea worm infection, as the worm does not infect individual intestinal cells. Primary response often works in conjunction with the innate immune response, and many of the immune cells that are active in the innate immune response are also active during the primary response. Macrophages and dendritic cells during the innate response engulf and destroy any foreign pathogens that are found in the bloodstream. These cells also present antigens on their MHC2 molecules, which then help the T and B cells to carry out a more effective primary response. After the larva is destroyed the first time the human is infected with it, the immunological memory of humans is ready and prepared for other infections. When the previously infected human drinks water from the contaminated water source again, the water fleas, along with the larva, get ingested by the body. When the larva is lodged into the interstitial tissue once more, the innate immunity still attacks the larva. The macrophages and mast cells release histamine, the eosinophils release destructive enzymes, and the complement system binds to the molecule again to attract notice of the larva. However, this time around, there are memory B cells and memory helper T cells also present at the site of infection that, once triggered, rapidly clone into effector cells. The effector B cells work to produce more antibodies against the antigen present on the larva, and effector helper T cells replicate and produce cytokines that aid the function of B cells. Memory cells also clone into more memory cells for future protection against the larva infection.